In this video, we will discuss about human macrophage populations present in multiple tumors and related to the immune checkpoint inhibitions. In the previous video, we have understood that late-stage activation of the macrophage populations are related to the macrophage populations that are found in the patients who are treated with immune checkpoint inhibitions or immune checkpoint therapy. So they wanted to see whether those late-stage macrophage populations are present in multiple tumors or in other tumors or not. So in order to check that hypothesis, they did their analysis on the breast cancer data that is publicly available. So what they did is they used this Mulder clusters and check the overlapping of these uh, murine 6,3 signature genes over this Mulder cluster. Murine 6,3 C3, not 6,3. Murine C3 signature genes or murine C3 cluster genes are the genes which shows a late stage activation of the macrophages that we have discussed previously. So when they compare or when they overlap this Mulder cluster with this murine C3 genes signatures, what they have find out is that this Mulder cluster 6 is closely overlapping with these C3 signatures. And when they did the further analysis on this Mulder cluster 3, what they have seen is that in the healthy populations they see a less of this late stage, these macrophage activations populations, but in the case of this breast cancer, they can see here the analysis can be compared with the breast cancer and the breast healthy tissues. So we can see that in the breast cancer they see the more populations of this cell killing and whereas in the case of breast cancer healthy they see the less populations of the cells that contributes populations of the macrophages that contributes to the cell killing, whereas in the case of breast cancers they see the more populations of the macrophages contribute cell killing. And these are these anti-tumorigenic or pro-inflammatory macrophages. So thereafter they also did this GSEA analysis to find out the pathways and that are upregulated in this Mulder cluster 6, and they can see that the cell killings pathway in differentiations are more upregulated compared to the other regular pathways, that is cellular homeostasis. And then what they did is they have presented for sufficient evidence that Mulder cluster 3 have similar signatures of this murine C3 populations. Then thereafter what they did is they did this Tisney plot of this further patients samples, and they are able to convert segregate them into the 6 different populations that can be seen here. And thereafter they treat the patients with this immune checkpoint inhibition therapy, and after that they check the responded cells and the non-responded cells, and they can see that the responded cells are seems to be more, and they more closely align with this cluster 0 and to some extent with this cluster 2. And when they check this results with this Mulder cluster 6 signatures, or when they overlap this patient response with this Mulder cluster 6 signatures, they can see that the cluster 0 is more closely overlined, and to some extent cluster 2 is also overlined, but cluster 0 is the maximum that overlaps with the Mulder cluster 6 is we can say the cluster 0 is the maximum that overlaps with the Mulder cluster 6 signatures. And thereafter they check the patients with this cluster 0 and did the GSE analysis, and what they have find out is that in the GSE analysis the pathways that are upregulated related to this inflammation, cell killing, and whereas the normal pathways are not that much upregulated. And thereafter they after that they check this murine C3 populations. uh, so in this case what they did is that they here they compare it with this modular cluster 6 signature genes, and in this panel K they compare it with this murine C3 signature genes. So here also they find the similar results, that is cluster 0 seems to be more closely aligned with this murine C3 signatures, and murine C3 signatures has been extensively studied in the previous results, that murine C3 signatures corresponds to the late stage activation of the macrophages. So these comparisons further substantiate their point that this cluster 0 or the patients that are treated with patients that respond to this immune checkpoint has late stage activation of macrophages that they have observed in the previous studies, in the previous experiments that they have presented in this paper. And after that they also check this percentage of macrophages in different clusters, and what they have find out that patients who respond to their these therapies such as immune checkpoint inhibitions has more macrophages macrophages in the has more macrophages in the cases where the patients respond to this immune checkpoint therapies, both in the pre-treatment conditions, and also during the treatment conditions. That is when the immune checkpoint inhibitions is process is going on, or when the immune patients is under this immune checkpoint therapy course, or even before the immune checkpoint therapy course they see a increase in the macrophages, whereas the patients who are not responders they do not see that much significant increase. So this indicates that the patients whenever get treated or whenever get respond or whenever responds when patients whenever responds to this immune checkpoint inhibition therapy, in most of the cases they will generate the macrophage populations, that is late stage activation of macrophages that can contribute towards tumor suppressions. And in order to do so they need to have this CCR5 ligands that can attract these macrophages populations. So this from these informations they made a point that if CCR5 ligands are present in the patients after this treatment of this immune checkpoint inhibitions, then they can then there is a high chance that the immune checkpoint therapies can work or help in these activations of the late stage macrophages that can suppress or regress the tumor progressions in the patients. This is the final points that the authors has presented in this paper. So I hope you understood this whole story about this paper that the authors has presented, and also these experiments that I have experimental results that they have presented here. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.